So where we left off, we have our white colonies on this petri dish, and we know that the white colonies have taken up the transgene within their bin19 plasmid because they're not able to break down the substrate in the excal because they don't have an active lac-shed gene producing the enzyme that breaks down the substrate. So we want the white E. coli colonies. We take the white E. coli colonies because we know that they have our transgenic bin19 plasmids. So we've screened the E. coli with the lac-shed system, but in order for this transgene to actually enter the host plant cell that we want the gene to be expressed in, we need we need the bin19 plasmid to be inside of an agrobacterium, because in order for the tDNA, the transgene that's contained within the tDNA between the left and right border in that bin19 plasmid, it needs the Vir genes inside the agrobacterium of the Vir operon system to actually transport the tDNA from the bin19 plasmid into the plant cell genome. So once the bin19 plasmid is actually in the agrobacterium, it's basically the exact same concept as we went over with the TI plasmids, to take the tDNA within the left and right border and incorporate it into the plant cell genome. So the bin19 plasmid is non-mobilizable, so it can't conjugate between species alone. We can't immediately take the bin19 plasmid in our screened E. coli that we took from the petri dish. We can't just take them and then conjugate it with the agrobacterium to get the bin19 plasmid into the agrobacterium. We need to do a thing called triparental mating. And when we do this, we basically conjugate our E. coli that we screened and extracted from this petri dish. We conjugate it with another E. coli containing a different plasmid called the PRK 2013 plasmid, which is what we call mobilization positive, while the bin19 plasmid is mobilization negative. Once the bin19 plasmid and the PRK 2013 plasmid are together in this second E. coli that you can see in the diagram at the bottom, because the second plasmid is mobilization positive, it counteracts the effects of the mobilization negative bin19 plasmid, so they're able to jump the species barrier and the bin19 plasmid can make its way into the agrobacterium. And then we don't have to worry about the PRK 2013 plasmid because it doesn't replicate, it just eventually degrades, and we don't have to think about it. Our resultant agrobacterium containing our transgenic bin19 plasmid is what we call an LBA4404 agrobacterium. So this LBA4404 agrobacterium that we got from the triparental mating now contains the transgenic bin19 plasmid that we put in it from the transgenic mating. Remember, we've got this transgenic bin19 plasmid from these white colonies. It's gone from the E. coli and the E. coli in these white colonies to another E. coli and then into the final agrobacterium. And then we also have the other plasmid that we talked about at the beginning, containing the Vir genes for the Vir operon system, called the PAL4404 plasmid, and together this bin19 plasmid and the PAL4404 plasmid make up the binary system. So we have our tDNA, we have our left and right borders, exactly the same as with the TI plasmid, and we have our Vir operon genes that are going to transport this tDNA into the plant cell genome. The only difference is when we're talking about the TI plasmid, that the tDNA was oncogenic because it had our auxin and cytokinin genes that were causing the unregulated cell division, as well as the OPAN genes. And then another thing, in the binary system, we have our tDNA and the left and right borders in a separate plasmid to the Vir genes. So they're in two different plasmids, which is why we call it a binary system, instead of how it naturally is with the left and right borders and the tDNA and the Vir genes all being together in a TI plasmid. It should be noted that in these LBA4404 agrobacterium, we remove their oncogenic TI plasmids, so they're not actually causing formation of these galls in the plants that we're trying to mutate. Well, in the plants that we're trying to genetically engineer. So obviously we don't want massive cancerous growth in our food products. So our agrobacterium, LBA4404, basically our end product, containing the transgenic bin19 plasmid and the PAL4404 plasmid, they enter damaged plant cells. So if we remember, before we're talking about when agrobacterium detects the acetosyringein produced by damaged plant cells, some of the Vir genes recognize that. You have a receptor, and then that receptor phosphorylates another gene, which stimulates the whole process of moving the tDNA into the plant cell genome. So we purposely damage leaf cell discs, at least in the case with tomatoes, by cutting the leaves of the plant into discs, so the plant cells produce this acetosyringein, which activates the Vir operon system. The active Vir genes in the PAL4404 plasmid then get activated, and they transfer the tDNA within the borders of the bin19 plasmid that we've been talking about this entire time, our transgenic bin19 plasmid, into the plant cell genome. And these, when the agrobacterium infect these plant cells, they're going to incorporate our transgene that we've put inside of our transgenic bin19 plasmid, inside the tDNA within the lactase system like we've been talking about, inside the plant cell genome, and then the plant cell is going to be able to transcribe and translate and produce our desired gene product. We have to first separate plant leaf discs that have been infected by the agrobacterium from those that haven't, and we do this quite simply. We use selected plates containing an antibiotic called kanamycin, and our bin... Oh, I wrote that wrong. Running out of time. The bin19 plasmids contain a CAN-R gene, which gives them resistance to the kanamycin, which basically means that if the leaf discs have been infected, they'll be able to survive and grow and produce discs within 2 to 4 weeks. But if they haven't been infected, they'll just get bleached and die. And if we take a look at our original picture of the bin19 plasmid, we can see at the bottom right, we have our CAN-R gene there. And this CAN-R gene of the bin19 plasmid is what is giving these infected leaf discs resistance to the antibiotic in this stage. So it's important that you watch all of these so you get the full